Hi, my name is Vivian Dutton, and I did research this semester under Dr. Habeck. We were looking at the predictors of seed removal rates by invertebrates in a post-agricultural woodland. So plants tend to entice animals to disperse their seeds. Dispersal is the movement of a propagule to a new location. Many plant species have developed specialized dispersal strategies for their propagules. Fruits, for example, act as an attractive meal to many animals. The species pictured here are oriental bittersweet and pokeweed. These have fleshy pericarps that are attractive to many vertebrates living in Pennsylvania, such as birds and deer, also including squirrels, bears, fox, and more. These animals tend to eat the fruits, but the seed often survives undamaged. After the primary disperser eats the fruits, they tend to deposit the seeds in new locations. This is an example of primary dispersal, which is the first time a propagule moves to a new location. Animals deposit the seeds in new locations, which effectively disperses the plant's seeds. But the seed's location and fate still can change. Invertebrates tend to eat or secondarily disperse the seeds deposited by the primary dispersers. Secondary dispersal is the second time a propagule moves from the first location to a new location. There is lots of variation in preference for seeds, particular for plants. Invertebrates are probably cued by the costs and benefits of the reward. For example, if a seed is really heavy or chemically defended or hard to extract energy from, then the invertebrate might choose to leave the seed there. Whereas if the seed is smaller, easy to extract nutrients from, and is not chemically defended, then it would maybe be more likely to move that seed. We do know far less about seed dispersal by invertebrates than we do by vertebrates. But as long as the seed survives to germinate, the plant benefits. This is true for both native and invasive species. Seedlings often do better away from the parent plant due to a lack of competition, and also dispersal allows for establishment in previously unoccupied areas. So we wanted to explore some different aspects of secondary dispersal through soil invertebrates. These factors that we looked into included the species of the plant, species provenance, and also landscape orientation, which is the distance from the habitat edge into the forest. So according to the species of plant, as I said before, characteristics inherent to a particular plant's seeds could influence the seed removal rates. In regards to the provenance, surprisingly little is known about the potential disparity of the seed removal rates between species with or without an evolutionary history with local seed predators. And then finally, in regards to landscape orientation, edges tend to create unique physical and biotic situations which might influence the seed removal rates. But why is this important? The reason why we ask these questions is because understanding the rate of seed movement within a management parcel could give resource managers important information to reduce the influx or establishment of unwanted plant species, such as invasives. The area we chose to do our study was the Ronald R. Ryan Environmental Study Area. You guys might know it as the EcoPlat. This area is owned by Kutztown University, so it's a great resource for students to use for research and studying. It is a post-agricultural woodland. It was released from agriculture in 1975. It's currently dominated by invasive forbs and shrubs within the understory. And also, if seeds were predominantly secondarily dispersed from the edge to the interior, this would be useful information to know from a management perspective. So for the experiment, we chose 4 different species, 2 natives and 2 invasives. The 2 native species we chose were pokeweed and spicebush, and the 2 invasives were oriental bittersweet and mile-a-minute weed. These species were chosen according to the size of their seeds, but also each of these species can be found in or near the EcoPlat. So first we collected 250 fruits of each species, and the seeds were cleaned. This was done by removing the pericarp and allowing the fruit to soak in water, making it soft. and then by applying pressure to the fruit, the seed was separated from the pericarp. And then the seed was rinsed off and left to dry for 24 hours. In an effort to match up native invasive seed sizes for this study, spicebush was a large native which was paired with a larger invasive, mile a minute, and bittersweet was paired with pokeweed because they were also similar in size. So after each species were cleaned and dried, 10 of each were sorted into seed depots, which in this case were petri dishes. So that made a total of 100 different seed depots, 25 per species. After that, we made seed depot covers. These were constructed using Tupperware containers, and we cut 8-centimeter slits in either side of the Tupperware. And then on the opposite sides of those, we created holes using a large Bunsen burner and metal stakes we melted through the plastic to later stake these down into the ground. So after all the prep work was done with the seeds and the covers, the seed depots were able to be deployed. This is an example of one transect in the eco-plot. And there were 5 of these randomly scattered throughout the eco-plot, each starting at the edge of the woodland. Each transect contained 5 sample locations, and at each sample location, 1 depot is there per species. So the seed depots were placed at each location, covered, and then staked into the ground. So after the seeds were deployed on October 13th, they were counted every other day for 2 weeks until October 27th. The final seed count was taken from the initial seed count, and the averages for each species was calculated. Taking that average percentage of seed removal, we were then able to run some ANOVA tests, which are analysis of variance. So the first ANOVA we ran was a 2-way ANOVA of percent seed removal as a function of distance from the edge and species identity. This test found no significant difference between those factors. The second test we ran was another 2-way ANOVA of percent seed removal as a function of distance from the edge and provenance. Again, this found no significant result. However, species identity did stand out as a significant variable on its own. So we took that information and we conducted a Tukey's on a significant difference post hoc test to see which species differed from one another. And this is what we found. So letters that differ show that there was a significant difference in percent seed removal. Letters that are the same represent no significant difference. So as demonstrated in this graph, it was found that mile a minute was significantly different from the other species, whereas spicebush, pokeweed, and bittersweet were not different from each other, but only mile a minute. While doing this experiment, we learned that mile a minute has lysosomes. An lysosome is a fleshy protein and lipid-rich structure that's attached to the seed. This structure has been known to attract ants to aid with dispersal. So this could explain why mile a minute was the driving force behind the difference between species. It offered a potentially much greater reward when consumed than the other seeds that lacked the lysosomes did. So back to the questions we were asking in the beginning. Does species identity influence the rates of secondary seed dispersal? We found that yes, seed removal did differ among the species, and this was driven by mile a minute. Next, we asked if the secondary dispersal of seeds would depend on provenance, whether it was native or exotic, and we didn't find any supporting evidence for this. And then finally, we asked if landscape orientation, the distance from the habitat edge, would influence secondary seed dispersal, and we also were not able to find any significant evidence for this. In order to do this study, we did have to make a few assumptions. The first assumption was that cleaning the pericarp off the seed was sufficient enough to mimic the digestion of a primary disperser. The second assumption we had to make was that presenting the seed clean and instead of in waste would not affect the results of the experiment. And then finally, seeds could potentially be dispersed or predated. So for this experiment, we assumed that seed removal was dispersal. I'd like to thank Kutztown University and the biology department for allowing me to conduct this research. I would also like to thank Dr. Chris Habeck for allowing me to use his lab space, helping to plan and implement the experiment, and also providing the supplies needed to complete this experiment. Also, thank you to Dr. Dan Scavage for allowing me to use his lab space and tools in order to create the seed depot covers. Thank you to Dr. Chris Stackey for providing his knowledge of invasive seeds and the sizes of those seeds that can be found in the area. Thank you to Dr. Setliff for his help with the knowledge of invertebrate identification and also some experimental design. And thank you to those who helped with seed collection and seed depot deployment, including Kyle Bissett, Jason Denofa, and Jacqueline Race. Are there any questions? Yes.
Did your treatment of the seeds affect the lysosomes at all?
No, the lysosomes were still visible. Actually, if we go back to this picture was taken of one of the seeds after we had cleaned it. So that white structure, I don't know if this works, this is the lysosome right here. So it was still intact and present after we did the seed cleaning. Yes.
And did you observe ants, carrying these seeds at all or interacting with them?
We didn't specifically observe them carrying them, but we did find them in the area. Dr. Habeck and I actually did go out during this research study to a little haven sort of mile a minute within the eco-plot. And when we found the mile a minute on the ground, it was consumed. The lysosomes were consumed. We took those seeds that we found to Dr. Setliff, and he observed them under a microscope. And we did find some ant antennae inside of the seed, but we did not observe the ants specifically carrying the seeds, but we were able to find some species in the area interacting with the seeds. Any other questions? Yes.
So Vivian, do you think the seeds that were removed possibly by ants had a greater chance of germination? Do you think that they're influencing germination rates? And if so, how might you get at that question if you had to design another experiment?
I think they definitely could be influencing the dispersal from one place to another, but it depends whether the seed is actually being predated completely or not. If it is being predated, I don't think it will further germinate because the seed's being eaten. So further research needs to be done on exactly what the ants are doing with the seeds after they take them. So hopefully later on we could specifically find maybe an ant nest and observe them specifically with the mile a minute and test a specific instance of that. But further information would need to be discovered before we could do so. Yes.
Do lysosomes play a role typically in seed germination and enhancing the ability of seeds to germinate, or do they play a role in dispersal, or have they not been well studied?
I'm not 100% sure. I would assume they would play a role. They're there to attract ants, so it would make sense for them to help with dispersal, because if the ants are coming to get the seed because of this lysosome, it would make sense evolutionarily for the plant to be dispersed and hopefully germinate. So that would make sense, but I'm not 100% sure.
I just wondered,
Right.
based on research in the literature.
Yeah.
Any other questions? Great, join me in thanking you. Thanks everybody, we have refreshments